Good afternoon, everyone. My name is Pasakamoru Lama. I'm a first-year bioinformatics and system biology student. I would like to warmly welcome you all to today's seminar on AI in bioinformatics and system biology field. Our speaker today is Dr. Silas Srisawath. He is assistant professor in bioinformatics, director of the Center of Excellence in Computational Molecular Biology, and co-director of Center of Artificial Intelligence in Medicine Research Affairs, Faculty of Medicine, Chulalongkorn University. Please join me in giving a warmly welcome to Dr. Silas Srisawath.
โอเคขอบคุณครับเดี๋ยวก่อนนะผมไม่ได้ถามมาก่อนอันนี้ภาษาอังกฤษหรือภาษาไทยโอเคได้ครับ Okay, so I hope everyone in the Zoom can hear me. So today we're going to talk about the application of AI in bioinformatics. So we're going to talk about AI, but since I imagine that more than half of the audience probably don't have a strong background in mathematics. So today when we talk about AI, we're going to focus more on the interpretation of what the AI does. And maybe we will take a look at a little bit at how the AI works, but we are not going to assume any knowledge or equation. So you can rest easy on that. Okay, so my background is in mathematics. So today I'm working in the interface between mathematics and biology, but my original background is pure math. So I used to work with blackboard and chalk and prove all the theorem without a care in the world. But now I have to care about application. So that's a big career change for me. But so basically my lifestyle can be summarized in this four comma comics. So basically, I'm coming with mathematical knowledge and the computational thinking, and I want to ask the biologists or clinicians about that problem, and then try to solve it with computers, with equations. And oftentimes, when we realize that after maybe several months, we realize it's too difficult. But that's the way that we roll. Okay, so today's agenda, we can talk about how AI transform bioinformatics, focusing on the algorithms that you probably have seen, like sequence alignment, variant calling, protein structure prediction, how AI transform those processes. And we need to talk a little bit about other techniques in the AI. Like you might be familiar with unsupervised and supervised learning. We will see a little bit how that can help biologists in understanding about the cells, about the tissues. Like as biologists, like as scientists, you are not going to worry about the methodology behind AI, but you want to know how AI help you answer biological problem. And since we are talking about AI, we're going to talk a little bit about how AI understands human data, like image or language. How does it understand? And nowadays we have a lot of generative models. So we're going to take a peek underneath that a little bit to see what changed from traditional models to generative models. And at the end, I'm going to close with some not recent now, but it's a new application of AI, agentic AI, that allows people to do automated scientific discovery or doing more complex research. Okay, so let's start with the first topic, AI transformation. So what I see as AI transformation starts a long time ago, when people switch from qualitative experiment like running jails, looking at the band, like you can tell that the expression increased, but increased by how many folds, increased by how many units. Like we cannot tell exactly from the result. To when we have sequencing data, when we have high throughput assays, now we are able to quantify like the number of molecules, the expression level of the gene, like the copy numbers of the genes. We have numbers that we can use to calculate changes in biology. We can describe biology with numbers now. So that's the first step. If you don't have data, you don't have it in digital form, you cannot apply any computational techniques because you require numbers for that. Okay. So just to kind of gradually shift you from kind of techniques that you know to the technique that you don't know yet. So you probably are familiar with statistics. Right? So when we analyze, let's say, gene expression data, right? We have expression data from cells of patients between two groups. You can calculate the average, like average expression for patient in group 1 versus group 2. And you can calculate the fold change. The expression might go up twofold in the patient compared to the healthy control. So you already have some kind of computational thinking, quantitative thinking applied to biology. Instead of saying that the expression level goes up, you can say that this goes up by twofold. So you already use some computational idea. But when you have to make conclusion, or when you have to make discovery in biology, like who knows if twofold is high or low? If the expression goes up 1.5-fold, is that good enough for you to say that this gene is affected by the disease? Like, having fold change is very subjective. Like some people might like 2.5-fold, some people might like 1.2-fold because they want more genes. So statistics have a role here. Like when we transform fold change to p-value using either normal distribution or any distribution, we are trying to make subjective criteria like fold change into something that's more objective. Is it statistically significant? Is it much higher than random? We are using statistics as a tool to be more objective about how we analyze, how we describe the data. Even though it can still be hacked, you can raise the p-value by doing something. We are not going to talk about that. So this is probably the area that you are most familiar with, right? Where statistics comes in to biological data and help you make sense of the data, help you make conclusion about the data. But there are limits to statistical techniques. Right? Some data, like RNA sequencing, some of you might learn that it is modeled with a negative binomial distribution. But if you look at the distribution, like it's not a perfect fit. So when we use statistics to fit real world data, like biological data, we are making some assumption. We are making some assumption that our data, our cells, is following some theoretical model. And it doesn't have to be like that. So in some complex system, we no longer have a way to actually describe it. For example, like, let's say you want to study a transcription factor, and you want to know if this transcription factor recognizes a certain nucleotide motif, this is the binding site for the protein. You may learn in biology class that some transcription factor can recognize the Tata box, TAA motif. But we have to ask, like, how do people know that this protein recognizes this pattern? So once we know this pattern, you can design like a mutagenesis experiment that you can mutate A into G and then see that, oh, the transcription factor no longer activates the gene. So you can validate it. But when people first discover this, like, how do they know? So for example, nowadays we can do chip-seq experiment. We have a transcription factor of interest. We can pull down the DNA that are bound to the transcription factor, and then we can do sequencing. So we are turning an phenomena in biology that we cannot see with our eye into data that we can see on the screen. And if we assume that all the reads, all the DNA that we get, these are attached to the protein, to the transcription factor. So whatever DNA motif that the transcription factor can recognize must be here in all these sequences. But we don't know where. Like when we get a read, it might be like 100 base pair, 200 base pair. But the motif might be 8 nucleotide. We still have the problem of determining kind of which location in the sequence that the motif appears. This is not something you can do with statistics. Like we don't have any idea about the distribution of the motif, so we cannot fit that. So people have to come up with algorithms, like a procedure that once we have the data and we have some assumption that our answer, some kind of motif, some kind of a nucleotide pattern, have to be present in every sequence, or almost every sequence. Then computer science people can come up with an algorithm. If you are curious, you can Google git sampling. So it's a way that you can start from having no idea about the motif, and you can run the algorithm until it converges to a certain nucleotide patterns that match to each and every input sequence that you use. So this is somewhat similar to when you run BLAST. When you run BLAST, you have your DNA sequence, your protein sequence. You want to know what species it came from. You want to know which function the gene does or the protein does. You do BLAST, and you get hit, right? You get matches from the database. So these are top hits, like very high score, and some poor hits with low score. So you have the score for each. Like when you have the BLAST result, like sometimes you get 300 score, sometimes 200, sometimes 50. So these algorithms will always find for you whatever the best result it can find. If you use human DNA and you BLAST it against a genome from dogs, the algorithm will still find the best hit for your gene in dogs. If you search human gene in bacteria, it might not give you any hit, but if it does, it will still try to find the best hit. So when people come up with algorithms like bioinformatics, part of bioinformatics is about algorithms. So when we use algorithms to analyze our data, essentially we are trying to find the best answer, the best hit, the best motif. But when we found this answer, we actually have no idea if this is correct. This is the best explanation. This is the best motif. But who's to say that this is the real motif that protein recognize in biology? So knowing just the score, like how well this match together, or the raw BLAST score, it's not enough to tell you if this is trustworthy. You also have to have some statistics. So this, again, statistics also help bioinformatics to justify whether 318, is it high or is it low, given your database size, given your DNA length, given all other knowledge about biology. So algorithms help us operate when we don't have a good statistical model to identify the answer, which sequence will match, which motifs will match. Like we don't have a statistical model for this, but we can design algorithms that identify these patterns, and then we check it with statistics to get the p-value for the results. But at some point, like we run into problems, not problem, but situation like variant calling. If we study human genome and we identify mutation in the patient, we want to identify which mutation might cause the disease. We might have DNA sequence from the patient and from the healthy control. And when people try to interpret mutation in humans, because we cannot tell directly like what's the molecular impact for those mutations. Some mutation in non-coding area, we have no idea what it does. Even in coding area, some mutation might change the protein structure. Some mutation might do nothing at all. So people have to come up with several criteria to filter for mutations that are interesting. Because if you analyze a person's genome, you might identify 10,000 mutations, and we don't have enough resource to validate all 10,000 mutations. So people have to come up with like ways to filter out. If I don't have good base quality, like the nucleotide might not be trustworthy, then we want to remove it. If we don't have many reads, covering that mutation, then we might not want to trust it. If the allele frequency for that mutation looks weird, like in the population, many people have it, then we may assume that because many people can have this mutation and none of them are sick, so this mutation might not be interesting. So people have to come up with qualitative or criteria that allow them to read through large amount of data, like sequencing data. But at a certain point, like, we want to be objective about this. Like, we don't want one lab to say that, oh, I require a read depth of 5. Another lab says, no, I require read depth of 2, and they never trust it. So every lab has different criteria. Again, we have a problem of people being subjective. So one way that we can become objective again, this time, is to use machine learning. So the idea of machine learning is we're going to ask the computer to act in place of us. The computer is going to see sequencing data. And we're going to tell the computer for patients that we know the answer, which mutation caused the disease. So we have some patients where we know which mutation is the cause. When we treat the patient, then the disease goes away. So we know some answer. What we're going to do is we're going to ask the computer to do this process for us. The computer is going to look at sequencing data, and we can tell it for some mutation, whether it's the causative mutation or not. And the computer is going to learn what some kind of models here that act as our decision making. So this process used to be done by humans. We filter the mutation based on criteria that we think makes sense. We are trying to offload that to computers. If you see enough sequencing data from many people, if you know enough kind of mutation that cause the disease, can the computer make the decision for us? So if I summarize this process, so the way that we use machine learning to become our objective criteria is that we have a pain point. Right? We don't have theoretical model, we don't have statistical model to describe the sequencing data that we are seeing. And in many cases, we have too many parameters. Sequencing quality, read depth, allele frequency, whether it's encoding or non-coding, whether it's non-synonymous or synonymous mutation. There are so many descriptions, so many parameters. As a human, we cannot combine many parameters in our head. If it's coding, but it's low quality, versus if it's non-coding, but it's high quality, how can we weight these kind of parameters? We can. This is not something that human can do. So we try to offload this decision making by combining multiple criteria, by looking at the raw data to the computer. But before the computer can do that, right, we have to have data with answer. We have to have some data where we know the actual mutation for each patient so that we can tell the computer if they are making the right decision. So this is a process that we can train the computer to do. But one of the big questions is, as a bioinformatician, I think every one of you here is a bioinformatician in some way. We have the process that goes from sequencing data, the raw data, all the way down to mutation, which mutation to study further. We have the entire process. So which part would you want to offload to the machine? Do you want to offload the entire pipeline? Like we don't do anything. We just get the fast queue and then send it to computer program, and then we get the mutation at the end? Or do we trust in bioinformatics that we know, and then we only ask the computer to do the part that we cannot before? So we still have that, we can still make that decision, which part to use the computer. So doing this kind of seminar, you're going to see more and more techniques that allow you to replace different parts of the bioinformatics pipeline with computers. So I mentioned that computers can help us combine like multiple criteria together, which we are not good at. So we can even expand on that idea. So in addition to just one sequencing data, we can combine multiple sequencing data, and we ask the computer to look at multiple evidence, like not just the sequencing data from the patient, but maybe summary of allele frequency from the Thai population, like from Genomics Thailand, to look at them together before making the decision. So the original pipeline might be just identify the variant based on the sequencing data from one patient or from your cohort. But with modern techniques, we can also ask the model to look at a different kind of data, like allele frequency summaries from the population. So these two are kind of different type of data now. We have sequencing data from one, and then we have allele frequency from the population. We can combine these together, which is kind of not really possible because we don't have a statistical model that describes the allele frequency and the score that you get from the sequencing data. So another example that I want to show you in terms of we are transforming from algorithms, from statistics to machine learning. And then eventually I'm going to introduce AI. One of the classical examples is protein structure modeling. So I think all of you know about AlphaFold when it came out and it got the Nobel Prize. So luckily for me, I kind of worked in this field before AlphaFold came out. So I can tell you a little bit about the history of how this field can transform. So before we have AlphaFold, if you want to know the 3D structure of some protein sequence, like you have the FASTA file here with the protein sequence, the algorithms have to blast the sequence that you have against the database to identify similar proteins. And the hope is that among those similar proteins, some of them people might have solved the structure for. So if there are structure for similar protein, you can use that as a starting point to model your structure. You can just change some part of the structure to get the one that you want. But sometimes, when you are studying new species or entirely new protein, like when you search in the database, there is no relative, there is no similar protein, there is nothing with 3D structure. But still, people can come up with ways that, okay, if we don't know the nearby structure at all, we can do a lot of prediction. We can predict a whole bunch of structure, and we can do some clustering with the hope that, with the assumption that the correct answer, the one that are biologically correct, like we might produce a lot of similar solutions, similar answers along that line. Structure that are non-biological, like structure that are very weird, like the algorithm shouldn't produce a lot. But structure that are biological, the structure might produce a lot of variation of that structure. So by doing some clustering on the structure, you can remove candidates that are not likely. You can remove some of these candidates. And you get some clusters that have a lot of possibilities, a lot of similar structures. And you can identify the central or the representative structures of them. And you can use these, like the center for all these clusters, as a seed, as an initial structure to fine-tune or to optimize the structure further. And you can do this several times until at the end, you are converging to a few candidates, all with high quality. So if you go back like 10 years, maybe 7 years, and you run protein structure prediction, you're going to expect to get at least 10 predictions because you want to see the diversity. You're going to see all these possibilities. And you have to validate, either with experiment or with other evidence, which one seems to be the most, which one has the most likely to be correct. What happens when AlphaFold turn up? And this is not the current AlphaFold, but the first AlphaFold, when it's not called the headline just yet. AlphaFold version 1, if you are curious, I encourage you to read this paper. Not the newer one, but the first one. And the reason is, if you go back, like now that you know about bioinformatics, when you read this, you might be like me and you might laugh about how they decide things. So basically what AlphaFold did was they want to go from sequence, so every protein prediction starts from sequence. During that time, people realized that predicting 3D structure was still too hard. But one thing that they want to predict is the distance. Before we can predict the structure, we have to be able to say which residues come together in 3D, which residue that fold to meet each other in 3D. We want to predict this contact map, which residue in the structure are nearby each other in 3 dimensions. Because people know from earlier that if you have this information, you can build the structure really accurate. Because you have constraint that residue 5 might have to be close to residue number 100. That forces your model to bring residue number 100 close to number 5. So if we can predict this to the residue to residue distance, we have a good ingredient to predict 3D structure. And that's what AlphaFold version 1 did. So they take the sequence, perform the alignment, MSA, multiple sequence alignment. So basically they blast the input sequence against all protein with known structure. And what they use as an input for their machine learning model are the raw scores from these multiple sequence alignment. When you run bioinformatics, you get the e-value, you get the raw score, you get all the numbers. They just dump all those into the model. Even the number of sequence. If you blast your protein, if you get 100 hits, 100 is the input. If you blast and you get 5 hits, 5 is the input. So they can use everything that bioinformatics tools throughout into their model. So each of these layers that you see as multi-dimensional data, each layer is one output from bioinformatics tools. I think they even use multiple sequence alignment software to get a diversity of informatics score. And from those scores, they ask the model to do one thing, just predict if the two residues will be near each other or will be far away from each other. And once they have this prediction, they put it to a, I'm not sure if all you have experience optimizing protein 3D structure, but if you have this kind of constraint and the protein is a single polymer, so you have a polymer of protein of amino acids, and you have constraint that this residue has to be near this residue. You have this constraint, you can optimize the structure without predicting anything. You just optimize it where to place each atom. So what AlphaFold number 1 did, and this was still state of the art at that time, was to predict this matrix to be very accurate and use that information to optimize the structure. And if you go back to that time and read other papers that come from the same competition, there are many people that predict this matrix at the same accuracy as AlphaFold, but none of them perform this optimization as well as AlphaFold. So at that time, what I want to highlight is we go from a fully algorithmic and statistical techniques into the part where we offload the 3D distance prediction to machine learning. But we are still relying on bioinformatics to produce all the score, because bioinformatics tools, when they perform sequence alignment, all the diversity of amino acids at each residue, all the match score, these are information that the model can use to determine if two positions are related, are nearby each other. But still, most of the process is still based on bioinformatics and statistics. So this change in AlphaFold version 2, this is the version that got the Nobel Prize. And the key point that I want you to realize here, and you don't have to read the whole paper, is that AlphaFold version 1 only replaces one part of the process with machine learning. Only this part, from sequence to 3D distance constraints. The rest are still what we usually do. But in AlphaFold version 2, everything is all machine learning. And furthermore, they are mimicking what biologists think about sequence and structure. So in this diagram, so the input to the model, first the sequence. This is what every model sees, the protein sequence, and the multiple sequence alignment. So we have to understand related proteins. So within this workflow, so before like the sequence became 2D matrix of distance constraint and then became structure, it's a linear flow. But what, as a biologist, we would think that if we predict the 2D structure, the 2D constraint, and then we predict the 3D structure, one thing that we should do, we should check back whether this 3D structure is consistent with the 2D constraint. And is this structure consistent with the correlation between amino acids in the multiple sequence alignment? So what AlphaFold version 2 does is it flows all information all the way from start to the end. The sequence goes from the start to the end. The 2D distance, like the constraint, goes from the start to the end. And the 3D structure, once it's predicted, it's used to check with both the sequence and the 2D information. And then update the structure again, and then goes back and update the structure again. And they do this 3 or more times, 3 or more cycles. So at the end, the result is consistent from sequence to 2D constraint to 3D structure. And in that paper, they will show that if they take out some of these parts, the performance breakdown. So the transformation by AI here is the fact that we still retain the thinking of human, of biologists, but we replace all the calculation or the decision making with AI. So all these gray boxes, these are all machine learning models inside, something that we no longer understand but we still understand the process. We start from sequence, we get the 2D constraint, and then we get the 3D structure. We use the 3D structure to check back with both the constraint and the sequence and update the process, run this several times until we converge to the final answer. And that's the one that are so accurate. They got the Nobel Prize. So one thing that I want to emphasize here again, I'm not sure if this is too technical, but let's try. So if you are familiar with computer vision, when we teach computer to understand image, we want computer to understand image with two dimensions and make prediction from it. So we ask the computer to look at different region when you see this type of diagram. This means the computer model look at this region on the image and then make this prediction. Look at this region on the image and then make this prediction. So it's looking at each location, like human eyes, we focus at different areas. This works well for image because if you, I'm going to take analogies outside of biology. If you look at an image of a person, whether that person appear in the corner, in the middle, or in the bottom, is still a person. So the fact that that's a face of a person doesn't change wherever you put it on the image. But sometimes you want the context is important. Let me show this example. So I'm going to talk about if you think about the language model. So when the model tries to understand image, I mentioned that the face can be anywhere in the image and it means the same thing. But if we talk about language, this term, this word like booked, whether it appears here or elsewhere, the meaning can change. Because here is a verb and the subject is he. He booked something. But if it's at the end, it might be someone else, not this person anymore. So in sequence data, in sequence data, the location is important. The fact that this word appears at the second position can be important. So I'm saying this because every time that you read new AI paper nowadays, you're going to see this term, something former. In this case, it's an evoformer, but you probably have heard the term transformer before. So when we try to offload biological data to AI, the AI has to be able to understand the sequence, like amino acid sequence. It has to be able to understand the 2D distance constraint. So for the distance constraint, we can think of it like an image. So we can ask the model to focus on different locations. But for amino acid sequence, the fact that alanine appear in the second position or alanine in the 100th position, the function is different, even though it's the same amino acid, but the function, the structural information is different. So the model has to be able to distinguish between position as well. And that's why I want to introduce you to this concept. If you want to study more, the keyword is attention and transformer. But what I want to point out is when we look at certain information, look at this sentence, he booked a room at a hotel. The fact that booked is in this sentence gives you some information that there is an action of someone booking something, someone reserving something. The fact that booked appear at the second position gives you even more information that, oh, this because it starts at the front, that means this sentence is talking about someone doing the acting of booked something. And the fact that booked, the word booked, is present at the second position with he, with a, with you, with at, with hotel nearby, gives you the full information that booked here is for he, and what's being booked is a room and is at a hotel. So this is something that the modern AI are at the last level here. It can understand a piece of information in the context. When you start learning machine learning, for example, with linear regression, like some of you might have done linear regression before. When you have a coefficient for a variable, it doesn't matter where that variable appears. You just multiply it by 2, multiply it by 5, multiply it by -1. It doesn't care where it appears. So the difference between simple model that we learn in class And the AI that we see in the application today is the ability to distinguish between information by itself, information in some context, and information in full context. So this is what gives the AI the ability. So for the first, this is the first section. So we look at some example of how the thinking, like statistical thinking, machine learning, and AI can help transform the process of bioinformatics. We are turning from all human to all models, all machines, all machine. We have an end-to-end solution where we feed the raw data into the model, and the model do all the processing, all the checking, until it gets to the final answer. And one thing that I want to emphasize, and this means that you have an important role in the development of future AI, is every time we talk about AI, like we always talk about the success story, like this, where you have AI that have a very big breakthrough. But if you actually read the AI literature, and I did read a lot of AI literature, and you will find that many people, many computer engineering people, actually failed to make a good AI. And one thing that's because when we want AI to understand complex data like biology, if we don't have the knowledge from biology, we don't know that the multiple sequence alignment should be checked with 2D distance constraint and should be checked with 3D structure. Like if you try to make the model go straight from sequence alignment directly to 3D structure in one flow, it will not work. It will not work as well as AlphaFold. The only one of the key reasons that make this work so well is it mimics the thinking of biology. It encodes the knowledge in biology into the structure itself. And that's very important. So nowadays, you're going to see many people trying to develop a foundation model for genomes, for DNA. And what people are struggling with is when you try to go directly from DNA, like the mutation, and you want to predict a disease, it's a big jump. How could the model understand what's the relationship between a mutation, like on one chromosome at one position? How would that go directly to the disease? We have to think about biology. So people are thinking that when we have a mutation, it might affect the accessibility and might affect the structure of the chromatin, of the chromosome, which might then affect the gene expression. If the chromatin became closed, then the gene will not be activated. If the chromatin is open, then we have more gene expression. And then genes also act on each other. So what people are saying that is that now if you want to build an AI for genome, we need to look at all the genes together. We need to know how the mutation in the patient together change the 3D organization of that patient's DNA, the chromosome. How does that change in 3D structure of the chromosome change the gene expression? And how does that change in gene expression for all the genes act on each other to change the whole network, the whole system? So this is the new direction in AI for biology, where it's not just about throwing data into transformer model. People have to design the architecture according to biology in order to make it work. And that's because, so one good example is protein prediction. So even though nowadays we have an ability to predict the protein structure very accurately, but as a biologist, you have to be a little bit skeptical, like how do you know kind of which environment is this protein in? What's the pH? And what happens if the cell change into a different cell type? Does that affect this in any way? This prediction has no context. It doesn't take into account anything about the cells, about the environment where the protein is formed. So many people say that the next stage for AI in biology is to incorporate all the context, which cell type, which tissue, which environmental state, in order to predict the biological information more accurately. Another thing that people have explored nowadays is, in addition to predicting the structure, they're also predicting the uncertainty. So usually when you think about prediction, you get just one number or one answer. But as a statistician, when you learn statistics, we want to know like if it's 5 plus or minus 1 or 5 plus or minus 5. the confidence is different. So nowadays, people are trying to make AI that not only predict the answer, but also how uncertain or how certain it is about the answer. So in AlphaFold, one of the features is it's not actually giving you just one structure, it's giving you all the variants for the position for each atom as well in the result. So it tries to show you which part of the structure is confident, which part of the structure is not confident. Okay, so that's the first the first part. I am told I have 2 hours, so we can go over this flow. Okay. So for the next part, from my experience, even though what we see in the news, like most of the eye-catching applications are supervised machine learning. So basically, we ask the model to predict something. But in your day-to-day work, like as a biologist, most of the time we don't want to predict. Like we want to understand what happens in the cells. Like we are not planning to predict, like if we treat this, what would the change be? Like we want to understand how the treatment make that change. We don't just want to predict what will change. We want to understand. So for the second part, I'm trying to talk about techniques in machine learning and AI, but try to talk about it in the form that we will use as a scientist, as a biologist. So the first part is about unsupervised learning. So this is a situation that you will most find yourself in, which is when you don't have the answer. So let's compare. So usually for supervised learning, you have the data and you have the answer. This is an image of an apple. So you're training the machine, the computers, to be able to say that this is an apple. This is supervised because you know the answer. You expect the machine to say apple, otherwise it's wrong. But in your day-to-day work, like when you collect transcriptomic data, when you collect genomics data, you don't actually know what the labels are. You don't actually know like how many subtypes of the cells are there. Like you collect all the transcriptome, and you want to study what cell types are there. You don't know what cell types are there. So this is akin to the unsupervised learning mode, where we have the data from multiple cell types, multiple fruits, like in this example. What we want the computer to be able to do is tell us that in this data, there are probably 2 cell types. One that looks like green and round, one that looks yellow and elongated. We want the computer to help us identify these 2 cell types without knowing that there are 2 cell types, without knowing what the 2 cell types are. This is the area that we want to do day to day. And this is unsupervised. The computer has no answer to guide. It has to make decisions based on just the data. But we can still do this. There are many techniques that allow us to do this and do this very well. So first, every time that you see figures in papers, in any literature, it's always in 2 dimensions because everything that's on the screen, anything that's on the paper, only have 2 dimensions, X and Y. But we know that, for example, if a dot represents a cell, and these are transcriptomic data, we know that each dot has 10,000 of numbers behind. But you can still see this kind of scatter plot in 2 dimensions, just 2 numbers. And you can still make sense of how many groups there are. So one of the techniques in unsupervised learning is how to reduce the dimension. You start with data that fill your entire Excel, but when you want to visualize, you only have 2 numbers, X and Y. How can you do it? How can you go from 10,000 genes to 2 numbers and still have all the information to help you understand the biology? This is one of the miracles of unsupervised learning. And the other one is, once you're able to visualize the data, now you want to be able to group the data, to cluster the data, to identify if you have 3 cell types, or 5, or 6, for example. So let's go over this one by one. So first, dimensionality reduction. So you might think that why is this possible? If we have 10,000 measurements for each gene for the cells, how can we still describe the cell with just 2 numbers? Because we have 10,000 numbers. If we describe it with 2, we're going to lose a lot of data. Actually, we're going to lose most of the data if we do only 2 numbers. The fact that we can do that is, first, it's because when we want to understand the structure of the data, like we want to see two structures, two clusters, we want to see three groups, like what we want to see are the grouping. We don't actually care about what details these cells are. We want to see that they are grouped together. We want to separate them from each other. We want to separate different cell types from each other. And if we want to just separate them, you don't need the full information. And this is a very simplified example, but if you have two data sets that are cube, 3D cube, and they are next to each other, not overlap. If you want to distinguish between these two sets, you don't need the whole 3 dimension. You can just look at the cross section and it's still separated. You can just look at one edge and it's still separated. So you can go from 3 dimension to 1 dimension without losing the ability to distinguish that there are two data here. Another reason why we can do this, so imagine like when we collect biological data, like gene expression. If we do RNA sequencing, we are going to measure the gene expression for thousands of genes together at once. And we do that because we don't know which gene is important. We collect the data for every gene. But when you are studying a certain system, a certain disease, like a certain plant, you don't actually need all 10,000 genes to describe the phenotype. Only some pathways, only some genes affect the phenotype. So even though you start with 10,000 dimensions, the essence of your system is probably built in maybe 10 genes, 20 genes. So the computers can help you identify those genes, those dimensions that are important for your system. And you can go from 10,000 down to 10, down to 15, and you can still describe everything in biology. So if you're interested in computer science, there is a hypothesis called manifold hypothesis, and it's what we just described. The hypothesis says that if you have a complex high-dimensional data set, even though you have like 10,000 numbers to describe the data, they actually can be described with much fewer numbers and still retaining all the important details. So it's what we just described in biology. And this is more general. For any type of data, our hypothesis is that we can do this process. We can reduce the dimension without losing much of the information. And that's the basis of how we can do this process. So you might be wondering, like, how do we actually do it? We know that in principle that we can do it. But the fun things about computational is how to do it. We know in principle we can do it. How can we do it? So I'm going to show you this kind of general principle, general framework for dimensional reduction, and it's going to be applicable to every technique you encounter. So what we have as a most basic form of dimensional reduction is we have the original data. We have all the samples, we have all the features, maybe gene expression, maybe mutation, which is a lot of features, 10,000 of features, typically for biology. Let's say what we want to do. We want to visualize this data. Okay, the animation goes here. So let's say the goal of the dimensional reduction is we want to visualize this data on the screen. We want to show this data to others in a way that they also see that there are two groups here. One group with this expression profile, the other group with the inverse expression profile. We want to show this information, this two groups structure. What can we do? So to start with, like, we have the data. So one thing that we can do is, okay, if I want to show that there are samples that are similar to each other, grouped together, and also these five are similar to each other, grouped together. The first thing that we need to define is what is our similarity. How can you say that two cells are similar or two samples are similar? We need to have some concept of similarity. Might be correlation in gene expression profile, might be some other function. But once you define the similarity, what you can create from your data is this kind of matrix. So each row and each column will be your sample. So these X1, X2, these are your sample. And for each pair of sample, you calculate the similarity. So let's say this is higher is worse. So x1 and x2 are not similar. They get a score 3. x1 and x3 also not similar. Score 3. x2 and x3, oh, they are similar. The score is 1. So this matrix gives you a sense of which sample are similar to which sample. You can see from here that, oh, x2, 3, and 4, these are similar to each other, and x1 is different. So this information can kind of guide you a little bit of which samples group together, which sample group together. But to go from this to the computer screen, where we all have 2 dimensions, what we want to do is, we want to, so imagine like on the screen, like when you see something that's near or far on the screen, what your eye is telling you is Euclidean distance. It's a direct distance, the line in space. So let's imagine that if someone can tell you the answer that, this data point, you have to put them here. If someone tells you this, let's assume that some module has tell you that you can put this here. If you can do that, then you can also calculate the Euclidean distance between all the data points on here. So you also get this kind of matrix. And the distance might be different, like this is in gene expression with correlation. This is on the screen with Euclidean distance. But you can do it. On the data, you can do one calculation. On the screen, if you know where the points are, you can do one calculation. So the general algorithm for dimensional reduction is to make these two as similar as possible. If you can put your sample on the screen in such a way that when you calculate the Euclidean distance, it's looking so similar to the original similarity, then what you are seeing here is essentially representing the original information. What differs from technique to technique is how they identify, is how they perform this process. But essentially, every algorithm, like T-SNE, UMAP, PCA, not PCA, but T-SNE, UMAP, they're going to try to make the location of the sample on the screen or in any dimension you want. The distance between the sample is going to be similar to the distance between the sample in your original data. And this is how you preserve the structure, the similarity between the patient, between the cells, from the original Excel table onto the screen. This process. So I'm not gonna go into the algorithms of how this is done for each technique, but I just want you to know the names so that when you use it, you know what they are. So these are popular dimensional reduction techniques. Some of you might have seen PCA before, principal component analysis. Some of you might have seen PCOA, principal coordinate analysis. And in newer papers, you're going to see a lot of T-SNE and UMAP. So these have slightly different kind of assumptions, different algorithms, but the goal is the same. We want to preserve the similarity between the samples from our data onto the screen or onto any dimension that we want. Okay. And to show you some example, so this is the digits data. So people write the numbers and we take the image, so it becomes an image. So each of these is 17 by 17 pixels, so it's about 300 pixels. We have data set from 2 data sets, like different organizations collect their employees' handwriting. By performing dimensional reduction from 300 pixels down to 2 numbers with PCA. So first with PCA, we can distinguish the difference between USPS and the MNIST dataset without doing anything. We are not telling the algorithm that these are USPS, these are MNIST. We just throw in the data. But because the way that USPS collect data and the way the MNIST collect data is different, you can see that the handwriting for the USPS is slightly bolder, like the lines are thicker. For MNIST, it's thinner. This difference is enough for PCA to know that you have 2 different data sets in here. But it's not good enough, it's not powerful enough, PCA is not powerful enough to distinguish the number. So when you label the data point by the numbers, that should be from 0 to 9, they're still overlapping. So some techniques, it's good enough to distinguish the difference between the data set, but still not powerful enough to see the difference among the numbers. But let's say with a different technique, like with UMAP, you put in the same data, you transform 300 numbers into 2. Now you have, again, the same perfect separation between data sets, but this time you also have the clustering of the same numbers together. And that's because, like, even with your eye, you can tell that if the image looks like one giant circle, it's probably all 0. These are all 0. If the image looks like just one line, then it's 1, definitely. But the rest might be more difficult. So one thing that you might notice here is the orange are very well separated and well clustered compared to the others. And this would be number 1, so 0 and 1. So because 1 is so different from the other, because it's just a single line, even PCA can tell that this is one group of data, one number, basically. But for the other numbers, it's too difficult for PCA. But for the technique like UMAP, you can see the separation of most of the numbers away from each other. But you can still see yellow, red, and brown, they are still on top of each other a little bit. And if you look at what they are, so red is 3, start from 0. So red is 3, yellow is 8, yellow is 8, yellow is 8, and brown is 5. So these 3 are next to each other because, let me remind myself again, 3, 5, and 8. So 3, 5, and 8 are numbers that have a lot of curves. So the model still have some trouble distinguishing these 3. But otherwise, it can distinguish other numbers. And you have to remember that we don't tell them what the numbers are. The model, in this case, the algorithms, only sees the pixel in the image. And that is enough for it to come up with 2 numbers for each image to place all the same number next to each other. So with dimensional reduction, we can reduce complex data onto the screen while still giving us enough details to make scientific discovery. Okay. So let's look at some real biology example. So this is from single cell transcriptome. So each dot is a cell, and the similarity between cells is based on the similarity between in that gene expression. So use UMAP and t-SNE, both are able to place all the cells onto the screen, onto 2D, in such a way that the cell type are still separated, and the location of the multipotent progenitor is located in the middle, because this can differentiate into other cell types. So with just similarity, these techniques can give you visualization that allow you to make conclusion or make discovery. Another example, so in this paper, they want to understand how embryonic stem cells develop into multiple different types of cells. So they follow the same process. They visualize the data by mapping onto 2 dimensions, and they can see that embryonic stem cells are on this side. We know some marker for the embryonic stem cells. And then we can tell that the cell probably differentiate from the left to the right into different lineage. So this is a start. And once we are able to visualize this, we know that the process of differentiation goes from left to right. So we can ask at different locations on this map, which genes are upregulated. So we can tell among the embryonic stem cells, these genes are highly expressed. Among the intermediate cells, these genes are highly expressed. Among the intermediate cells, these genes are highly expressed. So we can make discovery about the markers, about the switch, the gene that can determine the differentiation phase by looking at the positioning of the cell on this map. So underneath, they're going to do more analysis than this, but the intuition is that when we are able to visualize multidimensional data onto the screen, it helps us understand and deduce the biology of the system. There are many more examples like this, so maybe I'll go faster. Okay, so in this example, so each dot is a patient, and they want to understand if this patient have some similar gene expression profile. So these are cancer patients, and we have the transcriptomics data for their tissues. And people want to know, like, if all the patients are different, like everyone has their own unique gene expression, or if this cancer exhibits certain characteristics that each patient will fall in either of the profile. And in this case, with clustering, by connecting patients with similar gene expression together, they can identify a group of patients that are tightly linked. So these patients in blue are so similar to each other, they are connected to each other. Patients in greens are similar to each other, they are connected to each other. So they can identify 4 clusters from the similarity in gene expression. Allow them to propose that maybe the cancer progress into 4 different pathways that lead to these 4 different gene expression profiles. Let me skip this one. So for this part, like, we talked about how the computer don't even know any label. Like, it only sees the data, the transcriptomes, but it can still visualize the data for us. It can still group the data in a way that we can interpret the biology. We can understand the biology underneath. So what can we do with supervised learning then? We were talking about unsupervised before, where we don't tell the computer anything. So with supervised learning, even though we don't usually use it for scientific discovery, we can still do so, like this. So, for example, let's think about transcriptomics again. Like when you perform differential expression analysis, you compare gene expression from 2 conditions, and you want to know which gene or which pathway is affected in each condition. When we do this kind of differential analysis and we got the volcano plot, the y-axis being p-value and the x-axis being fold change, and these genes are significant, upregulated, downregulated. We are doing kind of a univariate statistic, like we are analyzing each gene almost separately. But what we want to do, like, we want to know if some genes, like, they might be important only when in combination with others. You might have to have upregulation of one gene before the second gene becomes relevant. And you cannot do that. You cannot see that effect with univariate analysis. So that gene by itself might not be differentially expressed, but when another gene is upregulated, the expression level for the second gene becomes important. We have to do some multivariate analysis. So the good things about machine learning is you can do multivariate. You can fit all your data, like many columns, into the model, and you get a prediction. But the problem about machine learning is what people call a black box. When you put in these 4 characteristics from a person, and you get this output for a disease probability, we have no idea whether this 0.4 is because of his age, like more than 60, is it because of the gender, like female, is it because of the high blood pressure, is it because of the high BMI? We have no idea. So if you want to use supervised learning, which learn from input and output, like prediction to make prediction, if we want to use this for science, we have to be able to deconvolute this 0.4 output. Where does it come from? We want to be able to say that, we want to be able to analyze that this 0.4, oh, it's plus 0.4 comes from the age, right? So this is based on the old age, not that old, this later. And if that person is female, then the probability actually goes down. So this disease is probably dominant in male. And high blood pressure and high BMI only contribute a little. So by knowing this, like, we can check, like, we can make discovery that this disease or this treatment somehow is related to the age and to the gender, but not to blood pressure and not to BMI. So if we want to use supervised learning for scientific discovery, we have to have this process, which is called the explainability process. We have to have the model being able to tell us, or being able to dissect the model to understand which part, which input contribute to the prediction. If we can do that, then we can use supervised learning. For example, we can predict the outcome of a treatment in cancer patient by inputting all the genes. As a black box, we learn nothing. But if we can ask the model to tell us which gene contribute to the treatment prediction, then we can make some discovery that these are the pathways, these are the genes that we think are related to the treatment. We can do validation experiment afterward. So we need this kind of capability. So when people do supervised learning, to use some model to fit the data, if you use simple model like linear regression or tree model, like yes, no kind of model, these are inherently explainable. When you build a model, you have the coefficient, you have the tree structure, you know which genes are being checked and what expression level are being checked and what's the decision of the model. So this kind of simple classical machine learning model, these are already explainable. When you fit your data in, you can extract the coefficient, you can understand how the model makes the decision. But for something like neural network, like AI, we have no idea what it's doing inside. So people come up with several ways to dissect very complex model. I'm going to introduce some here, but we don't have to worry about the details. So first, it's called LAM, like the fruit. So it's a local interpretable model agnostic explanation. And the idea is this: we all understand linear model, but we don't understand AI. So what we want to do with this technique is we want to approximate AI with linear model. And you might think like, how is that possible? If we can approximate AI with linear model, why don't we just build a linear model? So what being done here is when we want explanation for our data, sometimes we don't want the explanation for the whole model. Like in the case of this prediction, we want the explanation about this output of 0.4 for this particular patient. We want to understand for this particular patient, why does the model make this decision? We are not asking about the decision of the entire patient. We just want to grasp on one data point. And if we only care about a certain location in the data, we can approximate that location as a linear function. So let's say imagine that we have an AI that made this decision boundary. So any patient or any cells in the pink area will be predicted as positive. Any cells, any patient in the blue area will be negative. If we want to understand the whole model, this is not linear. But if we want to understand for this particular cells, for this particular patient, what's the model used to decide that this patient is positive, this cell is positive? It's based on just this local line that separates the positive and the negative. So people come up with ways to approximate this part with a line, with a linear function. And then we can make sense of the coefficient on each of the input features, each of the input genes. Which gene contribute to defining this line? That would be the gene that defines that this patient or this cell is positive. So this is one way. We can simplify some local part of the model of the complex AI as a linear function so that we can make sense. The second technique, don't worry about equation. I lie when I say there is no equation. So this idea comes from economics. In economics, one of the things that they want to study is called game theory. So how people play with each other. So imagine if you have a football team and you win a match. You want to have some type of algorithm that says who contribute to the result. Is it the striker who got the goals? Is it the goalkeeper who prevent the score? Is it the defender who keep the other side from scoring? In game theory, people want to be able to assign contribution from each team member. So in the case of machine learning, in the case of AI, we can think of AI as a team. It's a team that consists of each input parameters, like each gene. When we input the gene expression, we predict, let's say, treatment result. Treatment result is the output, the score of the team. And the contribution comes from each gene. Each gene contributes to making that prediction. So using the idea from game theory, we want to apply the same concept to understand the contribution from each gene on the final prediction of the treatment or the disease probability. So yeah, I don't, there's no better description than this equation, so that's why I have to put it. But the idea is simple. We want to assign contribution to each person, to each gene. And the way that we can do that is, imagine that if you want to know if a certain gene contributes to the prediction, what's the easiest thing that we can do? We can just take that gene out from the data. And if taking that gene out change the prediction, that means that gene contribute to the prediction. But if you take the gene out and the model still predict the same thing, that means that gene doesn't really mean anything to the model. You can take it out and the model still behave the same way. So this idea is similar to that. They are going to take out a piece of data at a time and see if the model still make the same decision. So these two techniques can apply to any model, because you can take any model, you can approximate certain part of it with a linear function. There's no rules against that. For any model, you can take out part of the data and you can check if the model still behave the same way. You can do this principle to any model. So these two are most popular to understand modern AI. So let me summarize this part. There's a lot of keyword there. So I'm seeing when we want to study biological problems, we want to discover knowledge. Usually we are based on hypothesis. We do literature review. We have certain idea. We go collect the data and we prove the hypothesis. That's the hypothesis-driven technique. But with machine learning, we are moving to data-driven. We are collecting a bunch of data. We might have some hunch what is true, what is false, but we are collecting a whole bunch of data. And then we are using either unsupervised learning to visualize or to cluster, to generate ideas. We see the data, we see some clusters, we start to have ideas. If there are 2 cell types, 3 cell types in the data. And with supervised learning, we can build a prediction model, but then we want to understand how the model made the decision. So we need the explainability to understand which gene contribute to the prediction. And in several cases, so these techniques can help each other. So once you visualize the data, once you cluster the data, you can have some target to train the model. If you see 3 clusters, you might now train the model to predict the cluster, and you want to see which gene contribute to defining the cluster. And when you build supervised learning model, sometimes you have knowledge that you can go back and use to change your visualization, change your coloring of the data. Okay, so that's the second part. For the third part, I want to talk about how AI understands human data, like image and text. And I want to start from models that we all understand, like linear model. In linear model, we have input, like being different genes or maybe other measurement, multiplied by the weight, and then we get the score. So we know that this is easy to understand. It's just a line. We talked about 3 model earlier. So 3 model is like a decision. The model has to check for each gene what the expression level. If it's higher than something, that goes one way. If it's lower, then goes the other way. So this usually mimics the way that we think. For example, when a doctor can examine a patient, the first thing the doctors might think, what's the age of the patient? If the patient is older than 30, then he might want to ask whether the patient smokes. But if the patient is below 18 student, then probably don't need to ask about smoking. The answer probably no. But then we might want to worry about the weight, if the student has an obesity problem. So the question can change depending on the answer of the first question. So this is also another kind of model that mimics the decision-making of humans. The third kind of model that I want to introduce is kind of a neighbor-based. So this is something like when you think about previous experience. You might see similar gene expression before in a different experiment, and that reminds you that, oh, maybe my new samples also have the same phenotype or the same characteristic as the other sample, because they have similar gene expression. or if you see like patient with similar test result, like you might think that they might have the same disease as someone else who used to have the same result. So these are very simple models. We can explain it in like several sentences, and we all understood what they do. But the limitation of this simple model is, of course, like if the model is simple, it cannot capture complex mathematical relationship. It cannot capture very weird, very variable trend. The second limitation of all these models that we know quite well is that in order to use this model, some human, has to turn the data into numbers, because this input has to be numbers. All the gene expression have to be numbers. You cannot put in text. You cannot put in image. There is no definition for weight times the image or weight times text. So that's the second limitation. Because when you put in this data, you might be tempted to say that image is numbers. But if you transform this image into numbers, it's going to look like this. That doesn't make any sense. With human eyes, we can see that this is the microscope image of cells. But if we just look at intensity, if someone just tells you the intensity for each pixel, you don't see anything. So even though it's number, like the intensity, the brightness are numbers, by itself, it's not very informative. You cannot interpret anything from that pattern. So what changed this, like removed all these limitations, is first is the artificial neural network models. I think you probably have seen this before. So people want to mimic the way that we can make intelligent decisions. And we do that by having a whole network of communication between neuron cells. So we want to mimic that in computers. So imagine that we have calculators, like very simple calculator. But if we connect them together, one calculator, when it gets the result, it sends the result to the next calculator. And the next calculator calculates something, and then send the result to the next calculator. What people hoped back then was that if we do this, we mimic biology, we can get intelligence from numbers. We can have very simple calculation, connect to each other large enough, and we get something intelligent. And what I want to mention is that within each of these calculation units, it's actually very simple. It's a linear function. So if you have gene expression coming into this neuron, like this calculator, it's going to do a linear combination. One way times gene 1, way 2 times gene 2, add to each other. But the catch is, before it sends the signal to the next calculator, there's an activation function. So there's a mathematical function here that has to be applied to the linear combination. And this also mimics biology. When we have a signal in our neural system, if the signal is low, it doesn't send the signal to the brain because the signal is too low. You don't need to pay attention to that. But if the signal is high, then it activates the signal to be sent to the next cell. So this activation function mimics that process. So this function will look like this. If the signal is low, nothing will get sent to the next calculator. But if the signal is higher and higher, more signal will get sent to the next calculator. So this kind of mimic the biological system. And the amazing thing is, so this motivation looks like comes from biology. We are mimicking the aspect of our natural neural network. But mathematically, if you are interested, there is this theorem back from 1989, the universal approximation theorem. The mathematical terms might seem like an alien for you, but the essence of this is that with this kind of structure, artificial neural network structure, and this kind of activation function that looks not too complex, right? With these two ingredients, you can mimic any continuous mathematical relationship. Basically, you have a universal calculator that can calculate anything. So usually calculators, the normal calculator can only add, subtract, multiply, and divide. Right? The scientific calculator might be able to do trigonometry, do exponential. But artificial neural network with only just one layer, like just one layer of neurons, with this kind of activation function, can calculate everything. So this is one essence that people think that we found the answer. We cannot use linear model to capture nonlinear relationship, but with artificial neural network, we have the tools to model any nonlinear relationship, anything. With some catch, there's some engineering tricks involved, but theoretically, you have a model that can do everything. You don't need to look anywhere else. This is it. It can do anything. So that's the first part. We have the model architecture that can do anything. The second problem that we have to solve is how can we feed data from text, from image into this model so that it can do its thing. It can approximate any relationship. But we need to transform our data into numbers first. And we all know that if we do a naive representation, like we change man to 1, woman to 2, like boy to 3, girl to 4, this doesn't mean much. The model doesn't understand the relationship between gender or between age at all. It doesn't know anything from 1, 2, 3, 4, 5. So we want to do something better. For image, like I already mentioned, if we turn this into just intensity, just brightness, it doesn't mean anything. We have to be more clever. We have to be more clever about how we turn these into numbers. And if you have done some image processing or image editing, like in Photoshop or on your phone, like, we know that from one image, we can do a lot with it. We can blur the image. We can even identify the edges in the image. If you want to detect the face of the person, there are mathematical ways that we can do that on the image. If you are interested, these are called image processing. So basically, there's this operation called convolutional operation that you can apply to different parts of the image, and you can extract signals that sometimes make the image blur, sometimes give you the edge in the image. So we have some tools that we can use to extract information from image. But the problem is, if it's a human face, we have some idea what to do because we are familiar with human face. But what about cells? Like if it's an image of cells, how do we know which convolutional operation to perform to get the feature that can help us? Let's say maybe classify the cellular location, or maybe even determine the cell type. How do we know which operation, which numbers basically? So when we change the convolutional operation, what we change are these numbers. When we change these numbers, we get a different multiplication product, and we get a different signal. So we know some of these numbers for blurring. These are the numbers: 0.0625, 0.125, and we know the numbers to extract the edges: -1, -1, -1, -1, 8, -1, -1. But how do we know which numbers is appropriate to classify cell type? And we don't. And this is where machine learning comes in. We're going to feed the model many different images of a cell where we know the type. We might do some immunofluorescence staining to know which cell type it is, we know the markers. So we can train the model that this image, whatever you do, whatever convolutional operation you do, in the end, I want this to be cardiac cell, for example. It's not cardiac cell. And for a different image, you might want it to predict this is liver cell. So the model, what it's going to do is it's going to learn these numbers, like coefficient, like linear model learn the coefficient. Convolutional model will learn the convolutional coefficient using a large amount of data to learn all these numbers that should be multiplied to the image to get the signal. And the signal will be good enough for it to classify either cellular localization or cell type. And this combination of neural network design and convolutional operation give rise to convolutional neural network that can allow the model to extract information from image and learn how to do that by itself. It keeps updating the convolution coefficients until we find the one that works well. So up to this point, we have a way that works. But as a scientist, we want to be careful. When the model is able to identify the cell type, does it make the decision the same way that we make that decision? Does it look at the cells or the image in the way that is similar to us? Otherwise, we probably cannot trust it. If it can make the correct answer, but maybe it's looking outside of the cells, then how can we trust that? So people also want to understand what happens inside the model. So for example, how the model understands that this is a car. We have techniques that we look at different positions inside the model and see what information are being used. So sometimes at the earliest steps, the model only sees colors and edges at different angles. But at a certain point, the model starts to assemble all the colors and all the edges into geometrical objects, and eventually into something that resembles parts of the car. We can see the wheel, we can see the light, we can see the bumper maybe. And if the model can understand this much, then we trust that it can classify a car. If it can see the wheel, it can see the light, it can see the bumper, it can see other parts of the car, then we trust that with this information, even human can classify that this is a car. So this is something that we want to make sure that the computer understands. And these different details that the computer uses to capture this image, we call this representation. So representation is the sense that this is the data, but the computer turns this data into different aspects, different characteristics that represent the data. So sometimes people call this representation of the data. And the essence of modern AI is the ability that the model can look at any raw data and turn it into a representation that even human can make sense. This is the ability that the model does by looking at a lot of data and being able to make this representation. Okay. So for biology, we use this model a lot for any kind of image. We can recognize different parts of the cells, we can recognize different type of objects, parasite or anything on the microscope image. This is because we have the convolutional operation that can focus on different regions on the image and find the coefficient that extract the signal that can tell us about the biological aspect of that area. And just want to know that this convolutional operation that look at different positions on the image can also be repurposed for DNA data. If we think of DNA motifs, we want to understand which nucleotide patterns on the sequence that are recognized by the protein. So let's say we have DNA sequence that the protein can bind, coming from ChIP-seq, and the sequence that we generate randomly, we don't expect the protein to bind. We can train the model, convolutional model, to look at the sequence that the protein can bind and the protein and the sequence that we generate randomly, and ask, can it predict which sequence that the protein can bind? And we ask it to do with convolutional operation. So the model is going to perform convolution, finding coefficient that somehow detect the motif. And if the model can learn the convolution coefficient that can detect the motif, when we put in DNA sequence with the motif, it will identify this position and activate the signal. If we put in a random DNA, it will not find the motif, so there will be no signal. So we can use convolution for the sequence data as well, this way. But for sequence, for language, what people actually does is not like that. We use a different technique. So just want to introduce a little bit about how the AI understand language, understand human text. So when we think about human text, each word, like king, man, woman, we might think of it as just one, let's say one number, like one data point, not one data point, one number. But when we actually, if you have to explain king to someone who doesn't know about king, your explanation is going to have many different aspects of the king. For example, you have to explain that king has to be a male, cannot be female. you have to explain that king is someone with kind of high status, high social status. Right? So the word king itself, even though it looks like a single word, but there are many aspects of the meaning inside. Like man, right? You have to explain that man is someone with above certain age, like otherwise you call boy, and the gender is male. And it's a term about human, like we don't call other animal with man or woman. So even though these are just one word, in essence, there are many dimensions about this word. So what kind of representation that we want from word is multidimensional representation where each dimension captures a certain aspect of the word. So this second dimension that is high in king, right, and low in man and woman might be a representation about social status. Like it's high for king, but for man and woman they are equal. So this third one, like they are low in both three words. Right? So this third representation dimension might be about either it's living thing, whether it's animal or not animal, or maybe it's living or non-living. So these three are about living things. And the last vector, the last column here might be about gender. King and man are both male, and woman is female. So we want the model to be able to transform from one word into multi-dimension, where each dimension is meaningful in some sense. But how can we do that? Do we need someone to label? Like, do we need someone to explain the aspect of all the words in the world to the model? No, like we cannot do that. That's too much work. So what people did is, okay, we have the internet. We have a lot of Wikipedia webpage. We have a lot of blog. Like we have a lot of Twitter comments. Can we do that? Can we ask the model to learn by itself? So there are two ways that we can do this. The first assumption is if the model can transform words into vectors that are meaningful and related to the word, the model should be able to use those representations to predict something about the sentence. If we take out the word "set" from the cat set on the map, and we ask the model, you got the cat on the, can you tell what's the missing word? If the model can predict that, that means the model is getting the representation that are meaningful, can tell that the word "set" is a proper term in this sentence. Another way we might call, people call this mask language model. So basically we are going to mask one of the words at random. We're going to remove some of the words. And we ask the model to see the rest of the sentence and then predict what the missing word is. If the model can do this for all the sentences that we put in, so that means the model understands the language without us having to do any work. This kind of training is interesting enough for me to select here because usually when you want to train the model, you need to collect the data. You need to have the gold standard for the label, which patient has which disease, which gene has which function. You have to have a gold standard annotation. But for language, people just say, we have enough data on the internet. We just ask the model to predict the missing words. And if they can do this, like for the whole internet, if the model is actually good enough to understand human language without having to do any particular label. So this is something that's very interesting. We can get the model to understand probably the most complex type of data without having to do much work. And what sparked interest in this language model is originally, like when they do this, I'm not sure if the engineer have high hopes for this approach, but when they do something like this, like very simple training process, they found that the model can understand the representation of the word very well. For example, between house, cat, kitten, and dog, house is different. It's not an animal, so it's further away. If you plot the embedding, the representation on 2D, it's further away. Between cat and kitten and dog, like cat and kitten are the same animal. Kitten is just a small cat. The model even understands that cat and kitten are similar more than to dog. And even more surprising, people found that the model understands that
if you change man to woman, like it's a change in gender. It's implicit, like when you change man to woman, you change the gender. The model even understand that the vector that point between man and woman should be similar to the vector that point between king and queen, because that's also a change in gender. So people found that the model can realize this relationship by being trained like this. So that's the kind of the magical part. Like you're training a model without saying anything about the grammar or the meaning at all. Like the model just see how we use the language, and is able to come up with representation that capture the way that we actually use the language. Okay. So that kind of progress into large language model nowadays. We are not going into that. Okay. So in biology, like, this ability for the model to understand like image, text, essentially anything is important, because in biology we have so many different data. Like if you go to UniProt, a protein, you can get the amino acid sequence, like you can get the description like as a text, like what the protein does as a text. UniProt also linked to other databases, like you can get the tissue-specific expression, you can get the associated disease, you get all kinds of data. If you go to Interpro, like you get the protein domain annotation, also a different type of data. If you go to string, you get protein interaction, also different kind of data. But with our ability to train the model to represent different kind of data, we can adapt the approach to make the model understand amino acid sequence, description, like text description, protein ID, protein-protein interaction as a graph, as a network, can be fed into the model so that it understands. And inside the model, another thing that's very interesting, like when we think about different type of data, if we want to combine them into a single number, so let's say you see the protein sequence and protein description, you see the domain, you see the interaction partners, and you want to predict will this protein be affected by certain disease or certain treatment, you have to combine all these evidence into just one decision. That's something that's very difficult for humans, as we mentioned. But for computers, we can just say that whatever you get from UniProt and Interpro, and whatever you get from string, you just concatenate them into a longer vector, and then use that to make prediction. So you might think like, wait, how do we concatenate information from two databases that are totally different, just attach it to each other and hope that it will work? So what happened here is, because neural network can approximate any relationship, it's a universal approximator. So what happened here is that instead of having to decide like how the data are integrated, we just say that the neural network, you can do whatever you need to do in order to make these two representations able to be concatenated and still make sense. So this is something that I'm not sure if you take this well or not, but when I first learned about modern machine learning and how people just concatenate or add something together without any apparent reason, it bothers me. But now that I look back at it, like, and think in terms of engineer, not as a scientist, as an engineer, like if you have a universal calculator, like, you don't need to do anything. You just ask it to calculate in any way that's needed for the data to be compatible with each other. So let's look at another example. So let's say you have multi-omics. This is a common situation that you will face. You have gene expression, you have DNA methylation, you have microRNA expression. So you have 3 omics. You want to combine them together to understand the cell. Because these are different omics, if you have to write a model as a biologist, so DNA methylation might affect gene, activate gene or replace gene that can change gene expression. Right? So at some loci might affect microRNA, and then microRNA might kind of downregulate certain genes. So the network will be like very messy as a system biologist. As a machine learning engineer, they just say that, okay, I'm going to represent the relationship between genes as a gene-gene network. And the strength, the edges in the network, I'm going to use co-expression, like correlation in gene expression, or maybe methylation, correlation in methylation profile, and maybe correlation in microRNA level. You get a network, and you feed it to neural network. And the neural network's task is to do whatever calculation, whatever mathematical approximation it needs to make these into data that have the same dimension and can be added together. So this part, like this define all knowledge in biology. Like we want to do mechanistic modeling that brings all the omics together. But in terms of AI, because neural networks can approximate anything, any mechanistic model that you think in your head. So basically it's doing those jobs. You are asking the model to do any mechanistic linking of the multi-omics to generate the representation that can be added to each other, and that's it. So some people approach the problem this way. So it makes a good prediction, but it doesn't help you understand anything about the system. But this is the capability of the modern AI. You can ask it to understand everything and package it inside the representation. Just want to show you what the modern approach looks like. Not good for science. Okay, so I want to explore more on the concept of representation that we just mentioned. So we have seen mass language model, like where we remove a certain word. Like in this case, when applied to biology, we're going to remove some amino acid and ask the model to look at the rest of the amino acid and predict the missing one. So if the model can do this, right, the model understands how the protein in nature forms. Because we all know that the protein in nature doesn't, so all amino acid is not, the protein in nature doesn't consist of all possible amino acid combinations. You don't just generate 20 times 20 times 20 amino sequence and you get a natural protein. In nature, the evolution confines the space of possible protein. So what people want to do with language model on protein is that if you can train the model to predict the missing amino acid at any location, that means the model can understand evolution. It knows that in the current life on Earth, right, these are the possible protein sequences. So people can use this to train the model to understand proteins without labeling anything. They are not making the model predict any property of the protein. They are just asking the model to predict what amino acid would be appropriate in this position of the protein. And once you have that model, like the model that can understand evolution of the protein, it has a good representation of knowledge about the protein. Then we can use those models to train, to predict what we want, like 3D structure, for example. So this makes the process easier. So before, like if you want to train the model to predict 3D structure, like in AlphaFold, you need to have from sequence to distance constraint to the 3D structure, and you have to define the architecture that mimics biology. But what people found nowadays is they use the mass language model techniques, like train the model without any supervision, like just let the model predict the missing amino acid. They get a model that understands protein. And then they train that protein to predict protein structure. And it works well, almost as well as AlphaFold. So many people are moving in this direction, where we kind of offload the entire process of thinking to AI. And it works well. But as biologists, we should kind of always consider what mechanisms or what biological processes are being mimicked or are being learned in this manner. Okay. I might skip some of these because of the interest of time, but because I sent you, I think you got all the slides. So if you are curious, so these are some kind of state-of-the-art AI in biology that I think teach you something about how people use AI nowadays. Let me find the one that looks interesting. Okay, maybe this one. So this is the most recent work on genomics. So people start to think that we have conquered the space of protein, like we can predict protein structure with minimal training. So now people want to know if we can predict genomes. So similar to the protein. So this approach also removes some part of the genome and ask the model to predict the missing part. So similar to language model. Instead of amino acids, now it's nucleotide. But one of the challenges is protein is small, relatively small, but nucleotide or genome is very large. So engineering people have to come up with some ways to be able to input like 1 million nucleotides into the model. For protein, it's like 1,000 amino acids, like 10,000 amino acids. But for nucleotide, you want 1 million base pair. So you need to do some engineering. But once you are able to do that, so what this paper demonstrates, it's still not what I would say a final solution, but what it demonstrates is it trained the nucleotide language model on almost all reference genomes in the world, from bacteria to human. Without telling the model anything, without telling the model that this is coding sequence, like this is UDR, it doesn't tell anything to the model. It just asks the model to predict the next amino acid, the next nucleotide, for example, without any knowledge from biology. But what they found, surprisingly, is the model understands the coding region. So this is a gene, and this is the 5' UTR, the unclassified region. And what they calculate here is they try to introduce mutation onto the sequence. So we take the natural genome and introduce mutation. And they found that if the mutation occurs in the first codon, this is a start codon. If this codon is disrupted, you don't have the protein being produced. And they found that the model understood if mutation occurs here, the effect is very severe. So this yellow means the effect is high. And inside the coding region, the model somehow understands the codon being a period of 3. Right? In the last position in the codon, you can change nucleotide, and it usually don't change the amino acid. But for the first position, when you change nucleotide, you usually change amino acid. So the model actually understands that in coding region, you have a period of 3 in terms of the effect of mutation, but not in the UTR. And this happens even though the model is not told that this is coding and this is UTR. So this is something that we found that when the model understands enough about DNA evolution, it understands which location looks like coding region, which might be more concerned, which region are not translated, which might be less concerned. So this is some surprising result that people found with the latest model. Okay. Let me see if I should mention this. I add too many things, to be frank. Okay. Okay, let me skip this. So these are similar in concept to before. So for example, when we want to train a model, like to understand this image and to generate the representation for this image. If we have label, like if we have answer, we can say that please predict 2. But for biology, like when we have image of the cells, we don't have any answer. When we want to train representation, we can ask it to use this to reconstruct the original image. So we imagine that we have no answer at all. We just ask the model to look at one microscopic image, reduce it to 10 numbers, and use these 10 numbers to produce the original microscopic image. If the model can do this, this means that these 10 numbers capture the essence of this cell. These 10 numbers capture this enough that it can be used to recreate that data. So this is something that we do in biology a lot because we don't have the answer. But we still want the model to learn some representation for us. When we talk about dimensionality reduction, we also doing something like this underneath. But we are saying that from original data, we want to produce 2D image, position of samples on the screen. And in the process, we are reducing the amount of numbers, the amount of dimension of the data. the reason why I picked this topic is because in biology, sometimes we have confounded. When we treat the cells with certain molecular compound, like we have different cell type, different treatment. Sometimes like the experimental design cannot remove all the confounding. Different cell type might have different response to the treatment. Different drug might have different effect. We want to understand the biology without being confounded by these factors. When we train the model, for example, let's say we put in gene expression data, and we want the model to create the representation for gene expression data. And because we don't know the cell type, we don't know anything, we just ask the model to recreate the gene expression data back, the same concept as this. But one thing that we can add is inside the model, when the model creates the representation for gene expression and is able to reconstruct the gene expression. So this means that the representation is meaningful, it captures the transcriptomic profile. Inside the model, people found that we can also ask the model to do other things as well. For example, if we don't want the model to recognize different cell types, like we want to understand gene expression that are universal across, not cell type, across cell line. Let's say we have different multiple cell lines in the data, and we have transcriptomic from different cell lines. Each will have different baseline expression because it's from different cell lines. But we want to understand the effect of the treatment. So we don't want our model to learn the representation that captures the cell line. But because cell lines are in the data, the gene expression here is going to contain information about the cell line. So what people did, which is called the biasing technique, is we also asked the model to use the representation here to predict the cell line. And if we train the model in a way that it can predict the cell line, that's bad because this information contains details about the cell line. So what people do with the biasing technique is during the training, we're going to ask the model to be able to reconstruct the gene expression, but we want to ask the model to forget about the cell line. And we can do this because when we update the model, so let's say the model wants to increase a certain coefficient by 1, right, to predict the cell line, we just subtract 1 instead. So we make the model further and further away from being able to predict the cell line. So this technique helps a lot in biology because when you combine data from multiple studies, they're going to have different batch effect, different bias. And if we train the model on those combined data, the model can remember the data source, the batch. So we can use this technique to make the model forget about the batch. Whatever representation we are getting here is good enough to reconstruct the gene expression, but it's no longer enough to predict the cell line. So this is a technique that can help. And I'm going to skip this one. Okay, so the last part, I'm going to talk a little bit about generative approach that we are seeing nowadays. And the concept is simple. When we talk about AI that can predict something, predict whether the patient will recover or not, when we have that kind of a classification, prediction, it's called discriminative model. We want to distinguish these two groups of cells or patients. And it's simple. The model only needs to learn this line. If the model can describe this line, it can distinguish the patient. But if we want the model to generate a realistic patient or a realistic gene expression profile, the model cannot learn just this dashed line. The model has to learn the whole population. So with generative model, it's much more difficult because it's no longer just learning one particular surface or line. It's having to learn the whole distribution. What are real and what are what are natural and what are not unnatural. Let's say if you have to train a model to distinguish these two banknotes, right, it's really simple because they are so different. But if you want to create a model that creates a new kind of $10 bank or $10 bill, right, that looks so real that it can fool people, that's very difficult. So when we nowadays, like when we train a generative model, sometimes what we want is not the ability to generate. But what we want is that when we have a generative model, it actually can describe the distribution of the data. So this means the model knows about this, like the whole population. So we can learn something from the model there. A discriminator model, you can only learn the coefficient of this line. You learn nothing about the data itself. You don't know what the possible in terms of biology. You only see this line. Okay, so let me pick some of this. So some example, we have, let's say, microscopic data, and sometimes we want to label the protein or label the RNA to either classify the cell type or to diagnose certain disease. And we have to do this for every sample if we want to see the protein. But once we perform enough staining, people start to see that, wait, if we have enough pairing between microscope data, bright field image, and fluorescent image, can we train the model to generate the fluorescent image from the bright field in the new sample, new tissues? Maybe we don't need to stain anymore. We just ask the model to generate this image for us. And then we can understand the distribution of the proteins. We can classify the cell type from this. So people call this virtual staining. Instead of doing the actual experiment, we ask the model to predict the result of the experiment. And this technique can also be used to correct blurry image. When you take image and it's out of focus, usually you have to do it again. But with models that can help increase the resolution, even you have a blurry image, sometimes it's enough to have a model to improve the focus, get the image for you. So these are generative approach. It generates something that's not in your data, but it can tell from your data that this could be what you would get if you have the right focus. Okay. In medical, now people are exploring how to generate realistic patient radiological image, histopathological image. In this study, it's interesting because sometimes when you want to study, as a bioinformatician, we want to understand the change in gene expression, maybe throughout the progression of a disease. But we know that we cannot get a lot of samples from the patient or from the cell culture. Sometimes we only have data from 3 months apart. We only have sparse data. So this paper shows that they train a generative AI, a generative model, that can create a realistic transcriptomic data from a data set. And they use this data to impute the missing value. If you have a new patient or a new cell culture, you collect the data maybe at the start, in the middle, and at the end. You don't have the data for every day because that's very costly. So you use generative model guided by your measurement to impute the missing values, the missing time point. So you get the full trajectory of your transcriptomic profile. And you can then treat this as a real experiment. Like you imagine that, I collect this data. You collect some of it and you impute some with generative model. And you can perform bioinformatic analysis on this. Like do differential expression, do clustering, and you get some knowledge. And this knowledge, many times, turns out to be true because the generative model actually understands the transcriptomic profile of this disease. So when we use it to impute the progression, this is realistic enough that when we do statistical analysis or bioinformatic analysis, we get the conclusion about pathways, about genes that actually can be used can be validated and become biological knowledge. Okay, diffusion model may be too much. Okay, let's skip there. Okay, so let me end this. I think my time is almost up, already up. So let me end this with the newest way that people use AI. So we have seen how computers or machine learning or AI replace some parts of bioinformatics. And we have seen it replace the whole pipeline entirely. We have seen how people use it to understand different type of data and combine different type of data together. But those are still just one model. Each application is one AI. What people are doing nowadays is making multiple AIs work in tandem. How many of you knows about genetic algorithm? No, okay, but you know about evolution, right? So genetic algorithm is called genetic because it mimics evolution. So let's say you want to optimize your maybe yeast, like your culture yeast for certain maybe to brew your beer, for example. And you want to optimize your yeast to make better and better beer. You can edit the gene in yeast, right? But you don't know what to edit. We don't know which gene make good beer. But with evolution, right? If we engineer your yeast so that, for example, if you want a certain alcohol percentage in your beer, right? You engineer the yeast so that if the yeast can survive very well in that percentage of alcohol. I mean, we will get censored by using this example. So if you engineer your yeast so that it survives very well with that percentage of alcohol or sugar, evolution will drive itself. Like the yeast will mutate, will gain some mutation, and the strain that survive well, like produce the required amount of sugar or alcohol, like will survive. So you get selection, you get better and better yeast that produce the beer that you want, like, with the perfect combination of sugar and alcohol. So genetic algorithm is like that. We want to optimize certain things, and we want to optimize it in a way that mimics evolution. In this case, Google released AlphaEvolve, and the idea is we want to write a software that do something, let's say sequence alignment. And we want the software that do sequence alignment as quick as possible. So the speed is the phenotype that we want. If the program has high speed and it do the sequence alignment correctly, it gets high fitness. If the software did the alignment wrong, or if the software is very slow, it gets low fitness. So the idea is we want to assign higher chance that software that can do sequence alignment very well and very fast have a chance to be copied to the next generation. And software that do it very poorly will be deleted from the population. So we are treating scripts like programming script like living being, and we are letting it pass on their genetic code, exactly their script, their code, to the next generation if they do it well, and delete it if they do it poorly. But the problem is, like, we know evolution occurs on the genome when it changes the DNA. But how do we evolve scripts? Like, how do we change the code and still make it runnable and make it better? So what Google did with AlphaEvolve is representing the code, like learning the representation for the code and for the program, and optimize that code randomly, like adding new line, deleting some line, adding new parameter, removing parameters, and test the code on certain example. If the code performs well, then it gets higher fitness. If the code performs poorly, it gets lower fitness. And then the model, the AlphaEvolve algorithms, will select the code with higher fitness and then edit that further, edit the representation further to get newer and newer code. And it keeps running this until in the end, it gets a program that's sometimes much better than human. So if you read their paper, and they will claim that their script, like, start from scratch, from an empty file, it can create a program that can generate something much better than human code. So this is a way that we use AI to do even more complex routine. And if you're interested in this, scRNA.ai, so it uses multiple AI to work among itself, to come up with a research plan, to write a code to test the plan, and to evaluate the result of the code, and to update itself over and over until it gets the code, the result that match, that prove or disprove the hypothesis, and then use language model to write the manuscript. So it has the whole research conception to carrying out the research, updating it, finding errors, fixing bugs, all the way to writing the paper by using multiple models. in tandem. This similar concept is also found in the latest model from Google. So in this example, they want the model to be able to talk to the patient. So one of the interesting things is when the doctors have to examine the patient, they need to ask the question, and based on the answer, they need to adapt that question until they arrive with enough information to make the diagnosis. So the process is very dynamic. And usual AI, they are not good enough to know all the medical knowledge and be able to process the answer from the patient and match them together to lead towards certain diagnosis. So in this paper, Google kind of used multiple AI models to talk to each other, one acting as a patient, one acting as a doctor, talk to each other until the doctor AI can make diagnosis very accurately, and then use that as a model that can help the doctors in real world. Because now this model can suggest the doctor what to ask, given the knowledge about the patient that's already known. So we are seeing like multiple AI kind of working together. And in scientific lab, we are seeing robots that usually we already have robot arm. We can ask the robot to pipette the sample from one plate to another to move things in the lab. But usually that's programmed by human. We assign the routine for the computers to do. But nowadays, we are having like a fully autonomous lab where the computers have camera. It will look at the experiment, look at the plate. It will try to put some enzymes maybe into the cell culture, and maybe the assay is fluorescent signal. And the model will be able to look at the image, take the fluorescent image, and if the enzyme create a strong fluorescent signal, then the computer learns that this enzyme is maybe something that we want. And it tries to optimize the protocol further and further to maximize the fluorescent signal. So the model can learn to repeat the wet lab process by itself. And with human just techniques that please maximize this signal from the plate. We start to see this more and more. Okay. So the last part, like we talked about the good things about the AI, but I want to end with some caution. So there are two problems with AI that makes it very fragile. So let's say you train a model to understand all the dogs in the world. And one day, someone put in the image of their favorite cat. What do you think the model will do? Like it knows about all the dogs in the world. It can classify everything about the dogs in the world. But when it encounters the image of a cat, it will predict whatever who knows, right? Whatever dogs looks the most like this cat. So this is called an hour distribution problem. When you train the model, like no matter how hard you train, data in the future, there will be a certain point where the data is no longer similar to everything that the model has seen. And at that point, the model will break. The other thing is, when we train the model, right, we ask the model to do something correctly. And we look at the end result. For example, we ask the model to put the bricks together. The model might learn to put it like this. And it works, like the block stays like stable. But in real life, like when we use it, the model might solve these 3 items. And instead of putting the ball on the box like, to make it stable, the model might think that, when I train, I always see this combination, pink on blue. So the model will do the same thing. We'll put pink on blue. So when the model makes something correctly, if we don't know the reason, like this is important. Because if we don't know the reason that it's doing this because of the shape or because of the color, in practice, it can use color, which is not what we want. Right? We want it to use the geometric shape to make it stable. So these are situations that can make AI break very easily. Okay. And this is real example from my research. So we try to make the model to understand chain X-ray, very simple nowadays. So what we found is that we can create 2 model variants. These 2 have the same performance. If you ask like how accurate they are, like they're the same. And for this image, they even answer the same. So they are both correct. But when we ask model A, like, where is the image that goes into your decision? And model B, like, where is the image that goes into the decision? It's completely different. So this is really important that the prediction alone is no longer enough. With the complex model that we have nowadays, like, it can make the correct decision from very weird input signal, and they still perform the same. So nowadays, we want explanation for our model. And this is our last example. So I try to find, so usually when people talk about hallucination, it's a famous problem. And usually it's very easy to detect. Right? But in this case, I found a certain report that very interesting. So they asked the model whether this disease, like late onset Pompe disease, is it related to river in any way? The interesting thing is, at the time that they asked this, the answer should be no, because there's no publication that river is involved in this disease. But the thing is, these authors purposely asked this question because they are about to publish a research that show a link between river and this disease. So in this case, like, there is no way to tell whether ChatGPT or GPT at the time, is it so good that it actually know this link before any clinician, or is it coming up with this from thin air, and it happened to be correct? So mistakes from AI, it might, example that we see now, usually like looks easy, but without deep knowledge, it's actually difficult to distinguish whether it's correct or whether it's not. Okay, so this is my last word. I know that many of you like want to move toward AI, but as we mentioned today many times, it has its pitfall as well. As a biologist, we want mechanistic model. We want to understand the knowledge. The fact that we do rigorous literature review help us reduce false positives. But when we feed the data into machine learning or into AI, we have no control. We have no control over what it does. What hypothesis is being used, like what knowledge is being used, we have no idea. So in terms of scientists, we should think of machine learning and AI as a tool, not as an endpoint, not something you want to make, but it's something you use to help in your study. Okay. And people are agreeing that going forward, if you want to make AI that's good for biology, it should rely on biological constraints, like physical constraints, chemical constraints, like how AlphaFold 2 incorporate the idea from human inside the architecture to make the state-of-the-art prediction for protein structure. We shouldn't try to do it in a way that is completely black box, but works very well. We don't want magic, we want knowledge. Okay, so that's everything I have for today.